Okay, so in today's lesson, we're going to look at immunofluorescence here. Right? And this idea here, immuno comes from immune system, and what we're talking about there are antibodies. Fluorescence is the idea that certain molecules, when put under a certain type of light, in this case, usually UV light, they will start to glow. Okay, and scientists use these two concepts to study molecules in biology. Now, you've already probably, or somebody looked at fluorescence, and that's the idea of tagging things. This is a bit more specific, and we'll get into the differences later. So it comes down to the basics of, right, antibodies will target specific antigens, okay? So this is used in your immune system. This is how you fight off infections and things. Now, scientists have co-opted this system, and what scientists can do is they can make specific antibodies for almost any molecule. Okay, so this is monoclonal antibodies, which is something you'll learn about later on. But the idea here is that this antigen can be anything we want. If we're studying a certain type of molecule, we can make antibodies for that molecule. And then what they do with that antibody is they attach these molecules here that then allow it to glow under UV light. Okay, so let's take an example here. We take the influenza virus. It has specific antigens, right? And this is a basis for influenza tests. If you test for influenza A or influenza B, each one has different antigens. And scientists have created these molecules that fluoresce and they attach them to these antibodies. Okay? They incubate them and then they will fluoresce under UV light. In our case, they change colors, but if you're a scientist and you're studying it more specifically, you might take that influenza virus, infect it with some cells like this, and see where those cells are at. So you can see here, you have the green cells there, right, are where the antibody attached. So what this would tell a scientist, for example, is for some reason the influenza is attacking the cells around here, here, and here. Okay, so it's a very useful piece of research. You can look at this right under a microscope. Now, what are the general differences between general fluorescence and immunofluorescence? General is really just, it's more general, right? It's less specific. So you can look at just tagging DNA, not a specific part of DNA, just DNA, organelles, and other parts of the cell. Immunofluorescence targets specific molecules, right? So you can look at their location, and in a lot of cases, the amount, how much they fluoresce. So here's a really good example. This is rat retinal cells. I'm not 100% sure what they're studying, but in the cyan color, so that's this green color here. You can see it all over the place. That's this filament protein called GFAP. Okay, so that is using immunofluorescence, right? That's an example of that. It's target specific. Now, the other one here says the cell nuclei in magenta, right? That is these around here, and that's going to be less specific, right? That's going to be using, you're just looking at to see where the nucleus are at. So notice the nuclei aren't staying with this protein, but these proteins seem to follow a specific pattern, suggesting those might be neurons, I suspect. So that somehow maybe that protein is involved in neuron formation, right? So that might be how you use this, okay? But the biggest take-home message here is general fluorescence is less specific. DNA, organelles, parts of the cells, while immunofluorescence stains are target specific. You're looking at specific molecules.